All right, hi again, everybody. Where were we at? Here we go. Okay, so I'm kind of thinking about the best way of starting this off. So remember how when we talked about the structure of a eukaryotic gene, you have a 5' UTR, you have exons and introns, then you have a 3' UTR. Right? 5' UTR is obviously very important for translation and so on. 3' UTR, also. However, that 3' UTR is also really important in stability. And the reason for that is that it contains the targets for, these microRNAs and particularly microRNAs. And so really what is going on here in kind of a general sense is that you need some way of identifying which mRNAs need to be degraded or shut off, right? Because you can't just like go, ooh, you can't look around the cell, right? There has to be some mechanism for identifying them. And that's essentially what these microRNAs provide, right? And so they are little bits of mRNA that are used to scan the mRNAs in the cell, and anywhere that they match, so literally like a barcode scanner, anywhere that they match, that's their target. So if you want to turn off an mRNA by degrading or inhibiting it, you need to express a microRNA that matches. Does that make sense? So you have to produce the barcode scanner, right, that's going to recognize a particular sequence in that target mRNA. And so that's the general kind of overview, big picture. The detail is how that's done. So essentially what what you produce is a small piece of double-stranded RNA. That's dsRNA. In there, you'll find the, I guess, scanner sequence that will recognize the target mRNA. But on its own, it can't do anything, right? So you can produce this little bit of double-stranded mRNA, and that's not going to do anything on its own. Step 2, process that dsRNA into smaller chunks, essentially 18 to 25 base pairs. Come on. And that's carried out by DICER. So its job is to recognize double-stranded RNA and then cut it up into small chunks. Step 3, degrade one strand, which is called the passenger strand, to leave behind the guide strand, i.e. the scanning bit. What are you doing, you batty dog? Step 4 is find your target mRNA using the guide strand. Steps 3 and 4 are carried out by the RNA-induced silencing complex, also called RISC. Good question, Adam. These steps are the process by which you can degrade mRNA or inhibit translation of an mRNA. Now, which you get depends on how well that guide strand matches. Perfect match. RISC cuts the target mRNA. And that basically results in unprotected ends. And that then leads to RNA's degradation of the target mRNA. Target mRNA no more, right? Can't be translated because it doesn't exist. So imperfect match. And I think from memory, it's like 3 base pairs wrong. Instead, results in risk binding to 3' UTR and preventing translation. And again, this is a specific process that targets particular mRNAs, right? It's not going to affect off like non-target mRNAs. It's not going to shut down expression of genes that it's not supposed to. And that specificity comes from the sequence in the microRNA. Which is also a gene that can be expressed. So again, you have another layer of control over this. Any questions? Hey, shush. Shush. There you go. Nothing's happening. And so what's really cool, and this is what I use for my research, is if you can introduce a double-stranded RNA that matches the mRNA of a gene that you want to turn off, you can actually turn off that gene yourself. So if I want to knock down function of a specific gene, say something involved in lipid synthesis, is kind of one of the things that I work on. I just have to feed my worms bacteria that express that double-stranded RNA. And those worms will go, oh crap, this isn't good. Let's shut this down. And they'll actually knock down expression of that specific gene. And so without mutating that gene, doing like mutation screens and all kinds of crazy stuff, you can actually turn off gene expression on a gene by gene manner, which is really, really neat. And they've actually done that for, I think it's something like 16,000 genes in C. elegans, which is crazy. Not me, that's way more work than I could do. But yeah, you can do some really cool stuff with this. Anybody got any questions about RNA silencing? Cool. Okay. So, and finally, like the last step in the whole process is once that protein has been made, now you can modify it after it's been made. And those modifications, some of them are just, intrinsic to that protein. They'll always happen to allow that protein to work. Quite often, things like that uh, are particularly the attachment of sugars, so glycosylation. A lot of the proteins that are on your cell surfaces have sugars attached to them. Others need to be cut or cleaved so that the bit remaining is functional. Insulin's a classic example of that, right? The pre-pro insulin that's produced by translation has to get cut twice before it's functional insulin that can actually activate the uptake of glucose. Other modifications can be conditional, right? So you can add them depending on other factors. And those conditional modifications can change that protein's activity and location, which in some cases equals activity. Right. And the most common one of that is phosphorylation. So that basically means the addition of a phosphate group. So the addition of phosphate groups is done by kinases, particular class of proteins. Their sole job is to add phosphate groups, and they will add them to specific residues, for example, specific amino acids. And removal of those groups is carried out by phosphatases. So, and some of those kinases and phosphatases are themselves turned on or off by phosphorylation. So you can have what's called a kinase cascade. You turn on one kinase by phosphorylation, it then phosphorylates the next kinase, which then gets turned on and phosphorylates the next one, and so on and so forth. So just want to give you a couple of cool examples of post-translational modification. One is the role of phosphorylation in protein activity. The other one is the role of cleavage in protein activity. And this one's a particularly cool one. It basically stops you dying from blood loss when you get a scrape or a cut or something like that. So the first example is about phosphorylation. And so basically you have, so this is some work that a friend of mine, Michelle Castelletto, did in C. elegans, a worm, and this worm, which is a parasite of humans, Strongyloides stercoralis. And she was interested in a transcription factor. Now, transcription factors have to bind DNA to work, right? That seems pretty simple. So if you want that transcription factor to work, it has to be in the nucleus. That's where the DNA is. If you want to turn it off, you can take it out of the nucleus. It doesn't function anymore, right? And that switch on-off in the nucleus, outside the nucleus, is carried out by phosphorylation. And so in this case, phosphorylation results in the protein leaving the nucleus. I'm just going to be lazy and write phosphorylation as P'(N) because otherwise it takes a long time. So if you have a phosphate group, protein leaves. Removal of the phosphate group equals entry into the nucleus. And that means it's now active. So now you're not affecting the protein's activity directly. It's not changing shape, so it's now functional or it's not. You're changing its activity by changing where it is in the cell. And these are really pretty pictures. These, oh, what do you want? These are really pretty pictures. These are green fluorescent protein reporter constructs. And so wherever you see green is where the DAF16 protein is. Right, this was actually a really groundbreaking bit of work because this was, I think, the first time this had been done in a parasite. Right, C. elegans is, common thing to do in parasitic nematodes. It was really wild. And so if you look at this closely, so this, I don't actually know what cell this one is, but this is the cytoplasm and the nucleus in that of a cell. Right, so that kind of round blob there is the nucleus, and that little dark blob is a nucleolus. All the rest of the stuff around is the cytoplasm. So normally you have a mixture of this protein in the cytoplasm and the nucleus, depending on what's going on. Now, if you get rid of the ability to phosphorylate that protein, that's what it's called a phosphonull. So you mutate the protein so that particular amino acid needed is not there anymore. You'll only ever see that protein in the nucleus, right? So that real bright blob. So these are two nuclei of two different cells. If you make it so that protein is always phosphorylated or acts as if it is, now you'll only ever find that protein in the cytoplasm. So this is a really neat example of how you can change location of a protein and change its activity as a result. And you can fiddle around with that if you want to, right? By changing different residues, you can actually manipulate what's going on. So that's a really cool bit of work. I like that. I think that was a very elegant experiment that she did. And it's also it's really funky because many proteins can be phosphorylated in more than one place. And so now the activity is the sum of all of those different phosphorylation sites. Some of them promote activity, some of them inhibit it. So whether or not that protein is functional, which is what we care about, depends on the balance of how those things are phosphorylated. And again, the things that phosphorylate them, the kinases and the dephosphorylate and the phosphatases, they're also controlled by all of these mechanisms that we're talking about. And final example is of cleavage, right? So all of you at some point or other in your life has fell over or cut yourself or, cut your fingers slicing up onions, or stuff like that. And so if that cut isn't sealed, essentially blood will leave and pathogens will enter, right? That's basically what happens when you have a cut in a capillary or blood vessel of some sort. Now, if you have mutations, particularly if you're male, especially in the clotting factors, you have what is hemophilia, right? And you can actually die from blood loss from something as simple as a grazed knee, right? Back in the olden days, before modern medicine, it was rare for hemophiliacs to actually reach adulthood. Typically, they would just by accident have some kind of scrape or injury, and then they would bleed to death as a result, which seems pretty horrible, to be honest. So anyway, there's a very specific response to a damaged blood vessel. And basically, first you need to kind of clog up the hole, right? Make the blood vessel smaller. You activate things which will kind of help those blood cells kind of form a plug. And then you mesh all of that plug together with something called fibrin. Now, fibrin is a part of a protein called fibrinogen, right? Which sticks to itself. Normally, it does not, because you can imagine you don't want your red blood cells sticking to each other in normal everyday life, right? That would be bad. You'd have blood clots up the wazoo and you'd die pretty quickly. So you have to very, very carefully control this process to make sure that it only happens when it's supposed to. And to do so, you have 3 different steps. First, you produce these clotting factors. That's produced by this damaged blood vessel. Then you cut the activator. This is called thrombin. So you cut and this is what, this is like a molecular model of thrombin, right? So in its inactive form, prothrombin form, it's part of this larger protein. When you have a ruptured blood vessel, that thrombin gets cut off and now is active, right? And its job is to cut fibrinogen so that the bits that stop it sticking to itself are no longer present. And when it does that, basically fibrin sticks to itself. And it'll stick to red blood cells, it'll stick to platelets, it'll stick to the wall of the blood vessel. And so that's what causes that clot to become permanent. If you've ever kind of cut yourself and then like flexed your finger afterward and it pops open, right? That's before this process has had a chance to properly stick everything together. And that allows your body time to kind of fix the injury and, basically prevent stuff getting into your blood that shouldn't be there. But it's a very carefully controlled process because you want it to happen very quickly and very effectively when you need it. You don't want it to happen ever when you don't need it to. So you produce blood clots that will then lead to either heart attacks or strokes, basically. So this is something you have to be very, 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 very careful to control. And the control is all done by protein cleavage, which is really neat. Anyone have questions? Questions? Let me open up the transgenic. So I just want to spend the last half hour or so of our last class just talking about some of the cool stuff that you can do with them, with this knowledge to figure out stuff. I already talked about RNAi a bunch, Let's see. Yeah, we can talk about that later maybe. But so there's two other really neat things you can do. One is gene targeting, right? So rather than randomly mutagenizing, yeah, an organism and seeing what mutations you get as a result, now we have the ability to very specifically introduce mutations that we want, which is an astonishing breakthrough. It's kind of a scary one too, right? We also have the ability to express transgenic proteins, right? So these are proteins that are not normally found in that organism. And so you've probably heard about transgenics in terms of crops, particularly maybe even animals, although I don't think there are many transgenic animals in production. But certainly in crops, the main ones are you express Bt toxin. So like essentially the same protein that you use in Botox, right, which kills insects. So any insects feeding on a Bt corn, for example, will then presumably die. Kind of a little bit different from injecting it into Hollywood stars' faces. That seems like a not such a great idea. But anyway, the result of doing that is muscle paralysis, which is why if you ever see people who've had Botox, they can't waggle their eyebrows, and they also can't smile very easily. Why either of those things you wouldn't want to do? I don't know. Perhaps I just don't care about having wrinkles. So, and I'm also not a Hollywood superstar. Not like, just in case you're wondering, I don't kind of after this go and make some kind of blockbuster film. Anyway, so another really cool thing, not yet. Hey, what's that guy's name? Oh, I remembered that film, Fifth Element. Can't believe I forgot it. It's my favorite film of all time. Yeah, Liam Neeson, I think. He used to be a carpenter for most of his life before somehow he ended up as a film superstar. Not entirely sure how he managed that. I'd like to know. Because then you could read about a genetics professor who becomes a film superstar. But anyway. So, yeah, I love The Fifth Element. Everything about it is just fabulous. Actually, they had a little bit of a tangent. I think it was Jean-Paul Gaultier, like the French fashion designer, actually designed the costumes from that film, which I think is really neat. Anyway, see you soon, love. So transgenics are super, super neat, right? So it allows you to ask questions that you wouldn't otherwise be able to do. And in many ways, this is Pepper, come here. I'm trying to stop our puppy from escaping. This is one of the reasons why I stopped working on parasites, because it's just so hard to do that work. The work that I showed you by Dr. Castelletto is like a rare exception to that. And so here you can see a transgenic worm is a transgenic bunny. What purpose you'd have for a bunny that glows green when you shine blue light on it, I'm not really sure. But one of the things that I've used this for a lot, other than my current work, is this project going back to this, right? Trying to figure out what these different proteins do. Essentially, I was able to figure out where they're expressed and what they, well, in large part, what they do, simply by tagging them with a green fluorescent protein. So this is a real cool thing. Also called GFP. Hey, quit it. Sorry, dog scratching at the door. That was originally from a jellyfish. Aquia Victoria from Australia, I think, from memory. And basically it's a protein that when you shine blue light on it, come on, absorbs the blue light photons and re-emits green light photons. And so you'll only ever see green where that protein is. And just as a, and you can tag this in all kinds of different ways, right? So this was the full genomic locus, like all of it. This is where I mutated the signal sequence that results in it being exported. So you can compare the two. And each of these individual promoters had their own protein GFP fusion. And so before that, we didn't really know where this protein was expressed, right? Or really even that there were 3 different proteins from the same gene. And so now you can see all of the places in which that gene is expressed in, right? So it's a really powerful tool to use to ask, you know, scientific questions, essentially. And again, that's another look back at our transgenic alligator embryo, right? So you can start asking questions on whole animal. And one of the really cool things about this is that you can do this on live animals. Obviously, this one probably isn't live anymore because it's no longer in its egg. But you can look at, you know, different genes being expressed in rodents, for example, where they're being expressed without killing that animal. So you can follow this over time. Or you can change the conditions in which that animal is in and see how the expression or localization or whatever of that protein changes as a result, right? So you can really literally manipulate things in a pretty fine controlled manner. And that obviously allows you to answer really cool questions too. But the thing which is the biggest game changer of all, hang on, where's CRISPR gone? Here we go, is CRISPR-Cas9. This is a genome editing. And so CRISPR is basically, and this is a cool thing, I actually had a friend back at Penn State who worked on this. She wasn't the one that's got the Nobel Prize, but she was working on it in another aspect. Really, it's basically a bacterial immune system, or kind of acquired immune system. So essentially, every time bacterium gets infected with a virus and survives, it keeps a little bit of that viral DNA in its own genome. It's like a little, I can't remember what CRISPR stands for. Something like palindromic, inverted palindromic repeat or something. I can't remember. There's the letters stand for something. Just can't remember what they are. So every next infection, oops, that bacterium compares its library with the infecting virus, and if there's a match, the viral DNA gets cut, well, destroyed really. And so this is a way that bacteria use to recognize foreign DNA and cut it. So obviously the first time you won't know what it is, you won't have a matching sequence. Every following time you'll be able to see it and you'll be able to kind of, kill the virus, stop it replicating before it kills you, right? So it's very much a kind of bacterial immune system. But what we've done, or what several very clever people have done, is basically modified that to our own purposes. And so there are really three parts to CRISPR-Cas9 gene editing. First part is the enzyme. And that does the cutting. That's easy enough. Second part is the target DNA. The bit you want to cut. And the third part, this is the really, really cool bit. This is how you tell the Cas9 protein which target to cut. And that you do that by using guide, single guide RNA. And this is the bit that recognizes the target DNA. Now you design the guide RNA. So if you want to modify or cut a piece of DNA in your genome, let's say, all you need is the sequence of that bit of DNA. And you design your guide RNA to match that. So if you add your guide RNA and Cas9 to a target cell, it will use the guide RNA to target or to find the target DNA and then cut it. And that's basically what's shown here. So these three parts, the target DNA, the guide RNA, and the Cas9 protein, are all part of the system. This PAM bit here is not like a type of anti-stick spray. It's actually a short sequence that has to be near the target DNA for this thing to work. And so what the Cas9 protein does is create a double-stranded break, or DSB. Now, you remember way back when we talked about homologous recombination, which occurs during prophase of DNA division and that's pretty not DNA, prophase DNA division, and also in like funky organisms that have more than one copy of their genome. So if you damage DNA, you have a double-stranded break and you don't have that around, you basically have to use what I call a Hail Mary DNA repair response, right? You have to stick that DNA back together. So if you have a DSB and no matching DNA, to help repair it. You have to use what's called non-homologous end joining, NHEJ. Which is basically a Hail Mary DNA repair, right? And it's not a very good one, right? And the reason being is that typically induces small insertion or deletion. So if you simply want to mutate a gene or a piece of DNA, adding your guide RNA and Cas9 to your target will result in that target being cut and a random mutation being introduced. Obviously, you don't have any control over what that mutation will be. It's just whatever happens as a result of the NHEJ repair mechanism. But the thing is, that's better than having, you know, 2 bits of your chromosome flapping around in the breeze, no longer stuck to each other, right? That's pretty catastrophic, right? So the non-homologous end joining does a job, it just doesn't do a very good job, right? So it prevents the cell from dying, but it typically results in mutation, right? So if you want to mutate something, you can do it that way. Or, and this is really where CRISPR-Cas9 took off, if you introduce a matching piece of DNA, that matches either side of the DSB, now the cell can use homologous recombination to repair the double-stranded break. And this is the really, really, really cool part about CRISPR-Cas9. As long as the bits at the side match, whatever is in between gets added into the genome. So really, it doesn't matter what's between those flanking sequences. It'll get incorporated into the genome. So now you can introduce, a piece of foreign DNA. You can introduce the same bit of DNA, but that you've changed. What else can you do? You can modify that gene in a very specific way. Right, so now you have really exquisite control over what happens. No longer is it going to be a random change that occurs. Now you can actually go through and change individual amino acids in a protein, right? Step by step by step. So you can engineer a protein. You're not adding like the whole gene in addition to what's already there. You're changing what is already there, you know, to suit whatever, you know, process or purpose you have. And so this is the bit that's both really exciting about CRISPR-Cas9. It's also the bit that's really scary about CRISPR-Cas9, because now if it's found that, well, essentially you can use this to solve disease, right? So there are trials going on right now. There's one recently. Shoot, what was that on? I can't remember what disease that individual had. Something that resulted in producing too much of a certain protein, which ended up being toxic or something, I can't remember. But also things like sickle cell disease are potential targets of this. So currently what you do is you take that individual cells out and you change them with CRISPR-Cas9. So in sickle cell disease, it's about introducing a neonatal hemoglobin gene, right? So you're not trying to fix the one that's there, you're just trying to produce another form of hemoglobin that isn't problematic. And then you put those red blood cell progenitors, hematopoietic cells, back into that person, and they will divide and produce the correct red blood cells. The future, though, is putting all this jazz into a virus. And so then you essentially infect that patient with that virus. As that virus gets into that patient's cells, it's going to carry this machinery, the guide RNA, the Cas9 protein, the donor DNA, and it's going to change the cells, all the cells in an individual, right? So this isn't something you have to do before birth or even to like specific subsets of cells, which you can extract out of that individual. The aim is to be able to do that in an individual. So potentially, if you don't like something about yourself, hair color perhaps, you know, you want to boost your intelligence. I'm not sure how you'd do that. But the idea is that you can now, well, will be able to modify your own DNA in the future. And that's really scary because A, we don't know what these modifications will do. Yeah, potentially the skin, right? Because your skin is generated from, your skin cells are replenished by stem cells, your skin stem cells. So if you can change those skin stem cells, you know, to produce perhaps more melanin, so you're less likely to get skin cancer, or any other changes, maybe to get rid of, you know, moles and stuff like that. Potentially that's a future application. Really, the thing that's scary about this is there's no real limit. If you can think of a modification you want to make to human DNA, there are now the tools by which you can do it. The only thing that's stopping us as a species or a society is the ethics of doing so. The fact that it's considered poor form to genetically modify humans unless it's to fix a disease. Even then, there's kind of issues around that. So yeah, it is very much, well, Marcus, there probably is. And one of them, not so much hidden, but there's a huge blow up a couple of years ago, I think, where a lab in China, like kind of like a rogue scientist, actually generated two girls that had been genetically modified. He changed the cell surface receptor called CCR5, which reduces your probability of getting HIV or being infected by HIV. So this has actually already been done. And thankfully, there was a huge outroar. right? And he got his work shut down and, you know, got punished as a result. I don't know if he got sent to jail or not, but there's really strong condemnation about it. But the techniques are out there, right? It's not kind of secret how to do this. So I'm sure there will be. I don't know. I mean, it's China. People often disappear when they do things that they shouldn't, and you never know what happens to them. I haven't followed up what happened to him. So, I mean, the scary thing is, Adam, is that the techniques for modifying, genetically modifying human embryos already exist, right? It's not, I mean, there's obviously challenges to it, but it's not really difficult stuff to do, right? So that's already in place. So technically speaking, if you could find someone who would do this work, you'd be able to genetically modify your children. Maybe make them glow green so you can find them in the dark. I don't know. The kind of the next level is to be able to genetically modify individuals that are already adults, right? So you can genetically modify yourself. That's a lot more challenging. But the basics of human genetic modification, they already exist. That Pandora's box has been opened. You can't put it back in. I'm not saying it's a bad technology. It's already producing enormous scientific advancements. It's just a very scary one because of all of the issues, ethical issues that kind of go along with it. Anyway, that's pretty much it from me. There's more information in here to help explain that. Basically, ending in this diagram here, which we went through. It's been an absolute hoot. I've really enjoyed this class. I think it's been a lot of fun having all the different questions and interaction and shared pet images and the like, including Mr. Roberts' fluffy, not fluffy cat, depending on which part of the cat you look at. He's probably looking around for it right now. But yeah, it has been a tough one. It's a tough course. Hopefully you've learned a lot from this, and really hopefully you've learned some of the just really cool aspects of genetics that there are. It's just astonishing. That's why I'm so happy to be a geneticist, right? Because there's always something to learn. There's always something cool that, you know, is just waiting to be discovered. Anyway, so that's it for lecture. Half an hour, we'll have the last lab. We're going to finish off the Eugene work. So if you have Eugene, fire it up. If not, try and get it quickly. I think most of you should have it already after last week. Pardon me. And we'll finish that work off so you can get that assignment done. And that and exam 4 are pretty much all that's left. So have a great lunch. I shall see you in lab in about half an hour.